All right, hello Cerritos Physiology students. Welcome back after your spring break and then our week off. I hope you guys are all doing well. This is the first lab in our online instruction series. We'll be using the PhysioEx exercise to skeletal muscle physiology. You'll be able to get to this under the My Lab and Mastering heading in Canvas, and then from there it'll lead you to PhysioEx. This lab report, you guys will have to turn in a lab report doing all 7 activities by next week, next Wednesday, April 8th. And there will be a quiz on April 8th. I'll open the quiz up at 8 a.m. Of all the material from this lab. All 7 lab activities sounds like a lot, but they shouldn't take you too long. It'll walk you through the lab. There will be some pre-lab and post-lab quizzes, but all together combined, these 7 activities that you'll turn in are just worth 7 points. Those 7 activities that you'll do through PhysioEx, the skeletal muscle physiology exercise, you'll need to save them as a PDF and then upload that PDF as a merged file under the Canvas page where it says assignments. Many of you know how to submit an assignment because you've been doing that to submit your Lab 9, the special senses. There was a file that I had attached to a previous announcement about PhysioEx instructions, and it takes you through how to open up PhysioEx, how to upload an assignment to Canvas, how to merge a PDF file from your lab report that you'll be doing from these PhysioEx assignments. So I'd encourage you to get familiar with that. And again, please let me know if you have any questions. So this lab that you'll be working with will be about skeletal muscle physiology. So you'll be learning and talking a lot about contraction of skeletal muscles between actin and myosin. We're not going to go into super detail here in lab. You'll get a lot of that in lecture with Dr. Tydell. But one thing you'll need to know for lab is what we talk about excitation, contraction, coupling, and that's the process by which any electrical stimulus given to a particular muscle will trigger the release of calcium by the sarcoplasmic reticulum, initiating the mechanism of muscle contraction by sarcomere shortening. I pull up this slide. It takes you through these steps of contraction or excitation, contraction, coupling. You'll see the neuromuscular junction is shown here at the top left of this picture, where the axon terminal, which is a part of your neuron, brings the stimulus to the muscle itself. It'll release acetylcholine, which is a neurotransmitter across the synaptic cleft, and that acetylcholine will bind to receptors and then start to propagate an action potential down the sarcolemma, which is just the plasma membrane of a muscle cell, muscle fiber. That action potential will travel down the T tubules, and when the action potential travels down the T tubules, it tells the calcium that's always stored in these blue-like structures with the processes sticking out. These are your sarcoplasmic reticulum structures, labeled SR in this picture. This is where calcium will be stored. The action potential, when it travels down the T tubule, it tells your calcium to be released from the sarcoplasmic reticulum. And calcium is what we need for contraction in skeletal muscle fibers. What is calcium so important for? Well, calcium, I'm on step number 3 here. Calcium ions will bind to troponin, and troponin changes shape, and it removes the blocking action of tropomyosin, revealing those sites where the myosin heads can bind to the actin. So right underneath step 3, you see this actin thin filament. It looks like a double wound bead structure. The tropomyosin is the thin like string which is wound between those beads, and the troponin is this little yellow bead. Normally the tropomyosin blocks the sites of the binding sites where the myosin heads will attach. But in the presence of calcium, calcium will bind to the troponin. Troponin changes the shape, and that moves the tropomyosin off of those binding sites. When those binding sites are exposed, then the head of the myosin, the thick filaments, can reach up and form a cross bridge or form an attachment to the actin thin filament. When it does that, the myosin head, which looks like it's right above the number 4 step, the myosin head looks like a little head of a golf club. When that attaches to the actin, it'll do what we call a power stroke and pull the actin past it to allow the actin filaments to slide past the myosin, and that will shorten the muscle and cause contraction of the muscle. Then calcium will be removed when the action potential is done. Calcium will go back into the sarcoplasm where it'll stay stored, and the tropomyosin will go back to blocking the binding sites on the actin. So this brings us to what a motor unit is, which you guys will talk about in the lab. A motor unit is made up of a single motor neuron and the skeletal muscle fiber it's innervated by. Groups of motor units can work together. We call that recruitment to coordinate the contractions of a single muscle. This is a look at what a motor unit looks like. You see the motor neuron coming up its processes where the axon will divide, will connect to the nerve muscle fiber that it will innervate, and we call that a neuromuscular junction. Neuro coming from a nerve cell, muscular because it's innervating a muscle cell, so we call it a neuromuscular junction. What we just looked at was what happens in that neuromuscular junction, how acetylcholine is released, and then it causes the release of calcium. So this is what a motor unit is. Your lab will also talk a lot about muscle twitches, and a muscle twitch is an involuntary contraction of the fibers that make up a muscle. Nerves control muscle fibers, as you know, and when something stimulates or damages a nerve, it causes the muscle fibers to contract, resulting in a twitch. A person can often see or feel these twitches below the skin, and I'm sure you guys might have experience seeing a twitch from your muscle. The 3 phases of a muscle twitch, we say there's a latent period, or the time from stimulation of the muscle until the shortening of the muscle begins. So this is kind of your lag time or rest time. Then there's the contraction period, which is when tension and shortening of the muscle occurs. And then there's the relaxation period, where the muscle goes back to its resting length. The time here is measured in milliseconds, and this graph shows a stimulus being applied, one stimulus to a muscle, and then how tension increases from that stimulus from the latent period, the contraction period, and then back down to the relaxation period. Again, this is measuring it in milliseconds, so this is happening extremely quickly. This is a little more detail about those 3 phases of a muscle twitch. The latent period is the lag time. It's the time from stimulation of the muscle until shortening of the muscle begins. So this is the time when a stimulus is applied until the contraction will start. The duration is about 2 milliseconds. During this period, depolarization occurs. That just means the action potential travels down the T tubules and causing the calcium to be released from the sarcoplasmic reticulum. During the latent period, the actin myosin will bind together, forming those cross bridges. The contraction period can last from anywhere from 10 to 100 milliseconds, and this is where you'll see the upward tracing on the graph, and this represents the tension and shortening of the muscle itself. So this is when the actin and myosin will slide past each other, the sarcomere will shorten, and the contraction will occur from the muscle. And then the relaxation period is when the muscle goes back to its resting state. This will be showed on a graph from the downward tracing. In this period, this is when the calcium is actively being transported back into the sarcoplasmic reticulum. This can also last between 10 and 100 milliseconds. Each muscle will differ in the amount of time it takes to get through each 3 phases. For example, your eye muscles can twitch extremely quickly, and we call that a fast twitch muscle, in about 12 milliseconds, whereas the gastrocnemius which is your calf muscle, if you remember from anatomy, can take about 36 milliseconds. Your soleus takes about 100 milliseconds, and those are both calf muscles. We call them slow twitch muscles. And if you remember slow twitch and fast twitch muscles, slow twitch muscles are generally found in areas of the body to help with endurance, to maintaining posture. Your calves are good at that if you're walking or standing on your feet a lot of the time. And fast twitch muscles don't need as much endurance, so they'll twitch much faster. So muscles can respond to changes in stimulus frequency, and we call these two different changes in stimulus frequency either summation or tetanus. And you'll be performing exercises on a muscle, talking about summation, tetanus, and then recruitment, which will be in the next slides. Summation occurs as successive stimuli are added together to produce a stronger muscle contraction. So summation is when the stimuli will be added together to produce a stronger muscle contraction. And tetanus is when these contractions become fused together to produce a continuous contraction. Obviously, it's probably not a good idea for your muscles to go into tetany if they're continuously contracted. Muscle response and changes to the strength of the stimulus applied. So we talked about muscle responses to the changes in stimulus frequency, and now we're looking at how muscles respond to the change in the strength of the stimulus. This is where we'll talk about motor unit recruitment, and this refers to the activation of additional motor units to accomplish an increase in contractile strength in a muscle. When we look at motor unit recruitment, we can graph it out, and you'll probably be familiar with these graphs after you look at your lab activities. This graph shows the tension and the stimuli which are being applied, and then the middle between the two graphs shows the proportion of nerve fibers excited. So what it's showing you is each muscle fiber is filled in with a yellow circle. That's showing how more muscle fibers are being recruited to help with the muscle contraction due to increased stimuli. So you see here the stimulus voltage of the stimulus itself, and how after a certain threshold, the tension of the muscle can only get up to about maximum contraction. So around 7, 8, and 9 motor recruitment, you'll see that all the nerve fibers have been filled in. So even though we keep adding stronger and stronger stimuli to that nerve, there's no more motor units that can be recruited. So we get what we call maximum contraction. So this is a look at what motor unit recruitment is. So that takes us to the end of this short PowerPoint describing a little bit of what you'll go over in lab. Again, you should complete all 7 activities, which will include pre-lab quizzes, post-lab quizzes, data reports, and review questions for each activity. It seems like a lot, but you'll see that once you get the hang of it, it actually will go pretty quickly. You'll save the document as a PDF. To do that, again, look at instructions under the most recent announcement. I will also upload that instruction file as its own separate file to Canvas. Then you'll upload that PDF to Canvas as a merged file under the Assignments tab. And there will be a quiz next week on this lab. So your assignment is due next Wednesday, and the quiz I will open on Wednesday at 8 a.m. I'll leave it open for 24 hours. It'll be 15 questions, most likely multiple choice. Good luck with this, guys. I know this is our first lab, so we're kind of working through this together. Hopefully in a couple more weeks we'll have everything ironed out. Hope you guys are all being healthy and well. Take care.